Okay, hello to all of you. Let's discuss chapter number 17. And chapter number 17 is basically about gene expression. So in this chapter, we are going to talk about the two steps of gene expression, which is transcription and translation. So in the chapter number 16, we talked about DNA replication, how DNA make its copy. And in this chapter, we are going to talk about the protein expression or gene expression, because DNA is basically information and the functional part is basically the expression of the protein, because it's the proteins which do the majority of the function. So proteins are the most versatile biomolecules because they are the ones which perform many functions in the cell. So basically, in the gene expression, the first step is transcription. In transcription, from DNA, mRNA is made, and the next step is translation, in which mRNA is converted into polypeptide chains, and the translation takes place on the ribosomes. Okay, first of all, we are going to talk about the differences between the gene expression in prokaryotic and the eukaryotic cells. So in the prokaryotic cells, as you know, that there is no nucleus. So the transcription and translation takes place at the same place. So the DNA is first of all converted into mRNA, and then this mRNA is converted into protein. So the ribosome comes and binds on the mRNA, and then the translation takes place. And also in the prokaryotic cells, the transcription is not finished yet and the translation starts. But in the case of eukaryotic cells, there are many differences. In the eukaryotic cells, transcription, which is the conversion, which is the formation of mRNA from DNA, it takes place inside the nucleus. And then that mRNA moves to the cytoplasm, and there it binds to the ribosomes, and there the translation takes place. So translation is the second step of protein expression, and it takes place in the cytoplasm. So there is difference, especially in terms of the protein expression in the eukaryotes. So in eukaryotes, transcription takes place in the nucleus, translation takes place in the cytoplasm. But in the case of prokaryotes, because there is no nucleus, everything takes place at the same place. There is no difference in the space for the transcription and translation. And in the eukaryotes, when the mRNA is made, that mRNA is not mature form of mRNA. It needs some processings. Only then that mRNA is converted into mature form of mRNA, and that mRNA, which is a mature form of mRNA, leaves the nucleus, goes to the cytoplasm, and then it is ready for the translation. But this kind of a processing, we will talk in detail about what kind of processings takes place in the mRNA. We are going to talk about that shortly. Those processings do not take place in the mRNA in the prokaryotes. So in the prokaryotes, no processing. mRNA is not even completely made. Ribosome will come and bind on the mRNA, and the process of translation takes place. So that is why protein expression is very fast in prokaryotic cells. In the bacteria, the protein expression is very fast as compared to eukaryotic cells, because in the eukaryotic cells, the mRNA which is made is pre-mRNA. It's not a mature form of mRNA. It needs further processing, and only then the pre-mRNA is converted into mature form of mRNA, and that mRNA moves to the cytoplasm, and on the ribosomes which are present in the cytoplasm, translation takes place. So these are the main differences between the protein expression or gene expression in the prokaryotic and the eukaryotic cells. So this is in the bacterial cell, and this is what is happening in the eukaryotic cell. So you can see this is DNA. It's first of all converted into pre-mRNA, and this pre-mRNA gets processed it gets converted into mature form of mRNA, and then this mature form of mRNA will go through these nuclear pores, will leave the nucleus, and it goes into the cytoplasm. And in the cytoplasm, there are ribosomes. It goes and binds on the ribosomes, and there the translation takes place, which is the formation of polypeptide chain. So the information is converted from DNA to RNA, and then from RNA to protein. This is called the cell. DNA to RNA to protein, this central. Okay, now, so as we just talked about central dogma, that the information from DNA is converted into mRNA, and then it's converted into proteins. So we are going to now talk about all these things. So basically we know that there are 20 amino acids, and amino acids are the monomers of the proteins. So there are 20 amino acids, and there are only 4 nucleotides. So how does this whole system works? There are 20 amino acids, and there are only 4 nucleotides in the DNA. How does the information is there in DNA is coded in the form of amino acids? So how are the instructions for assembling amino acids into proteins encoded into DNA? Okay, the flow of information from gene to protein is based on a triplet code. The flow of information from gene to protein is based on a triplet code, a series of non-overlapping 3 nucleotide words. So let me show you this picture. So basically this picture shows everything. So this is DNA. DNA, as you know, is double-stranded, and the 2 strands of DNA, they are antiparallel in nature. So one strand is 3' to 5', another is 5' to 3'. So as DNA has 2 strands, one of the strand of DNA is going to act as a template strand. So template strand is that strand which will be used as a parent strand, or it will be used as a strand, and it will be used, and an mRNA strand will be made on the basis of the sequence in the template strand. So mRNA strand will be made on the basis of the sequence in the template strand of DNA, and mRNA strand is made on the basis of the complementary base pairing rules. Another important thing to know here is transcription also works in 5' to 3' direction. So in chapter number 16, we learned that DNA polymerase works in 5' to 3' direction, which means it adds nucleotides at the 3' free 3' end, because at the 3' end there is a hydroxyl group. So DNA polymerase adds nucleotides at the 3' end. Now so in the process of transcription, we'll talk about in detail. There is a very important enzyme involved. It's called RNA polymerase, and RNA polymerase also works in 5' to 3' direction. So the direction of transcription is 5' to 3'. So which means that the mRNA strand which will be made will be 5' to 3'. So the template strand of DNA, is going to be 3' to 5'. Again, the 2 strands, although DNA and RNA, they form a hybrid complex. It's only for very short time, because this DNA and RNA complex doesn't stay for long. These 2 strands don't stay for long. DNA strand is only used as a reference strand because its sequence will be used, and the mRNA strand will be made on the basis of complementary base pairing rules. So this is, so it means that if the mRNA strand goes from 5' to 3', if the mRNA strand goes from 5' to 3', in DNA, the template strand is 3' to 5'. So this strand, so the DNA strand is, the template strand of DNA is 3' to 5'. mRNA strand goes from 5' to 3'. So this strand will be used as a template strand. So you can see here, if it is A here, A always links with T, but that is in the case of DNA. In transcription, mRNA strand is going to be made. In RNA, there is no T. In RNA, there is U. So instead of T, there will be U here. If it is C here, it's going to be G. C, G, A, U. So you can see that this is the way mRNA strand is going to be made. This is transcription. After transcription is translation. So in the translation, three nucleotides, sorry. In the translation step, three ribonucleotides, they form one codon. So very important thing to know, three ribonucleotides form one codon. So always remember, codons are always on mRNA. Okay. So three ribonucleotides form one codon. So UGG, this is one codon. UUU, this is another codon. GGC is third codon. UCA is the fourth codon. And every codon codes for one amino acid. Every codon codes for one amino acid. UGG, like here you can see UGG codes for tryptophan. UUU codes for phenylalanine. GGC codes for glycine. UCA codes for serine. So this is the way translation takes place. Okay. We are going to, so in this chapter, we are going to talk about in detail about this process. So first step is transcription, second is translation. Okay. Now this is called genetic code table. Okay. So there are 64 codons. Okay. There are 64 codons. And out of 64 codons, there are 3 codons which are stop codons. So stop codons means whenever those codons are there in mRNA, the translation machinery gets a signal that they needs to stop making the protein. That's called the stop signal or stop codons. So out of 64, 3 codons are stop codons and 61 codons, they code for amino acids. They code for amino acids. So you know that there are 20 amino acids, but there are 61 codons. So which means that there are more codons for one amino acid. So there is more than one codon. So it means that there is more than one code for one amino acid. Okay, so that is why we call genetic code is redundant. More than one codon may specify a particular amino acid. But no codon specifies more than one amino acid. So genetic code is redundant. More than one codon may specify a particular amino acid. So for one amino acid, there can be 3 codons. There can be 2 codons. But one codon does not code for 2 amino acids. I hope it's clear. Okay, so this is the genetic code. You can see there are 64 codons. So these are codons. UUU, this is 3 ribonucleotides. 3 ribonucleotides make one codon. Okay, so this is also called triplet code. UUU, it codes for phenylalanine. UUC also codes for phenylalanine. UUA codes for leucine. UUG also codes for leucine. Then you can see CUU also codes for leucine. CUC also codes for leucine. Okay, so there are 64 codons. Out of 64 codons, 3 codons are stop codons. So let's see what are the stop codons. UAA, UAG and UGA. So wherever in mRNA these codons are present, they give the signal to the translation machinery to stop making the polypeptide chain. Okay, so they do not code for any amino acid, but they are signal to stop the process of translation. And there is another codon called AUG. AUG is a start codon. So wherever this codon is there, it's a signal for the translation machinery to start the process of translation. So AUG is a start codon, and AUG also codes for an amino acid called methionine. Okay, so it means that the first amino acid is usually methionine. You can see this is the genetic code table. So there are Like for serine, AGU, AGC, they code for serine. So this is for glycine, GGU, GGC, GGA, GGG. They all code for glycine. So now, if you don't remember, you don't need to remember or memorize these codons, because wherever you will get the question, you will get this table. Okay, but you need to understand things conceptually, because you can have many questions on your exam where you can be asked, this is the codon, which amino acid is there in the peptide, or you can be given an mRNA chain, you can be asked to make a polypeptide chain. So that's what you need to understand. Okay. Yeah, very important thing to know is that codons must be read in a correct reading frame, okay, in order for the specified polypeptide to be produced. So now let me go back here and explain this thing here. Like if you see this, so UGG is the first codon and it codes for tryptophan. UUU, it's a second codon. It's code for phenylalanine. Then GGC codes, this is third codon. It codes for glycine. So UCA, this is the fourth codon. It codes for serine. Now this is how these codons are read. Okay. So this is the first codon, this is the second codon, this third codon, this is the fourth codon. And every codon codes for one amino acid, right? So this codes for tryptophan, this codes for phenylalanine, this codes for glycine, this codes for serine. So this is called the reading frame. This is called the reading frame. If there is any alteration, suppose instead of UUU, there is A, it's going to code for a different amino acid. Okay, the reading frame changes. Okay, or suppose this G gets deleted from here. Okay, if suppose this G gets deleted from here, then the first codon will become UGU instead of UGG. It will become UGU instead of UGG. And what will happen because of that? As a result of that, the first amino acid is going to be different. And then the second amino acid is also going to be different, because the second codon will become UUG. It will become UUG. The second codon will also become different. Third codon will also become different. This way, the whole reading frame will change and a different protein will be made. Okay, so this is a very important concept to understand at this point. So genetic code is redundant in nature, and genetic code is also universal in nature. So what does that mean? Universal means that this genetic code table which we just studied, oh, I'm so sorry. This genetic code table which tells us that which codon codes for which amino acid, this is followed by all the organisms in the biological world. There are only few exceptions, but 99.9% of the organisms in the whole biological world follow this codon table, which means that whether it's bacteria or monkey, donkey, cat, dog, whatever, if some protein is going to be made, the amino acids will be coded by the same codons in the whole biological world. So that is why genetic code is universal in nature. And the biggest advantage of that is we can move genes from here and there. And that is why genetically modified food is possible. So you can see a tobacco plant expressing firefly gene. So you can take the firefly gene from the fireflies and you can put in a tobacco plant. It is going to glow. You can take jellyfish gene and you can put it in the pig. You can see how they are showing this fluorescence. So because genetic code is universal in nature, you can move genes from here and there. You can take one gene from one organism and put it into another organism. You can take vitamin A genes, you can put into rice. You can make rice more nutritious. Okay. You can take the firefly genes, which that gene is there. Nature has given that gene to fireflies. You can take that gene from there and put it in these plants. The plants are also going to glow like the fireflies. They are also going to give this light. So this is the advantage of genetic code being universal in nature. Okay, the second part we are going to talk now is we are going to talk the process of transcription, and then in the third part, in the second part, so I will stop the lecture with transcription, and then I'm going to make a second video for translation, and we will be also talking about mutations. So transcription, as we just discussed, is the first step of gene expression in which mRNA is made, and the very important enzyme which is involved in RNA polymerase. As I said, this enzyme also works in 5' to 3' direction, and it is going to a very important thing to know about RNA polymerase is it does not need the primer. The way DNA polymerase needs a primer, RNA polymerase does not need the primer, and it makes the mRNA strand complementary to the template strand in the DNA. So DNA has two strands. One of the strand is going to act as template strand. I just showed you that. So basically, overall process of transcription is there are 3 steps: initiation, elongation, and termination. So you can see that this is the DNA. Okay, in the DNA there is a region called promoter region. Okay, so promoter region is that region in the DNA where RNA polymerase is going to go and bind. Okay, so promoter region is a region in DNA where RNA polymerase goes and bind. So this region, which is light green shown here, this is promoter region, and also there is a in the promoter region there is a site called start site, or there is a point in the promoter from where the process of transcription actually starts, where the first ribonucleotide is added, and that is called the start point. So this is the promoter region on the DNA. This is the start point, and this is RNA polymerase. Okay, so on the promoter region, RNA polymerase goes and bind, and then what is going to happen? When the RNA polymerase binds to the DNA, the small part of the DNA is going to open up. So you can see how the DNA is getting unbound here. Okay, it's not that the whole DNA will open up, only the part of the DNA where RNA polymerase is binding, only that part is going to open up. Okay, and then the formation of RNA transcript will take place, and then RNA polymerase will keep on moving on the DNA. Okay, it will keep on moving on the DNA, and you can see how these 2 strands of the DNA, they're coming back, they rewind. Okay, so RNA polymerase will keep on moving forward, and the 2 strands, they will come back, they will rewind, and you can see how this RNA transcript is being made, and it is made in 5' to 3' direction. So it is made in 5' to 3' direction. So RNA polymerase will keep walking on the DNA. Okay, RNA polymerase will keep walking on the DNA. It will keep walking on the DNA till it finds the terminator sequence, and there RNA polymerase will leave the DNA. The 2 strands of DNA will come back the way they were before, and the RNA transcript is made. And then if in the eukaryotes, it's going to be processed, and then it will go to the cytoplasm where it is going to be translated. It is going to be bound by the ribosomes, and then it's going to be so the ribosome, we will talk about that when we reach that point. So the ribosome is going to go and bind, and it's going to walk on the mRNA, and then the process of translation will start. So the DNA sequence where RNA polymerase attaches is called promoter, as I just told you. In bacteria, the sequence signaling the end of transcription is called terminator. The stretch of DNA that is being transcribed is called the transcription unit. So a little bit more detail. Basically, we will be now talking in more detail about the process of transcription which takes place in the eukaryotes, because there are some differences between the prokaryotic and the eukaryotic transcription. So the 3 stages of transcription are initiation, elongation, and termination. So as we just talked about promoters, so the RNA polymerase basically binds to the promoter. And in the eukaryotic transcription, there is also involvement of some proteins. They're called transcription factors. Okay, the transcription factors, they basically go and bind to the DNA. So this is the DNA double-stranded, and this is the promoter region. In the promoter region, there is a region which is called Tata box. Why it's called Tata box? Because it is rich in thymines and adenines. That's why it's called Tata box. Okay, so this is the typical eukaryotic promoter. This is Tata box. This is the start point in the promoter. Okay, and the transcription factors first of all go and bind on the promoter of the DNA. Transcription factors go and bind. Transcription factors go and bind on the promoter of the DNA, and they actually help in the binding of RNA polymerase, because RNA polymerase is the one which is going to make the transcript. So the function of transcription factors, which are proteins, is to help in the binding of the RNA polymerase on the promoter region. Okay. So this is called the transcription initiation complex. So once the transcription initiation complex is made, then the elongation of the RNA strand takes place. So RNA polymerase moves along the DNA. As I said, it untwists the double helix 10 to 20 base pairs at a time. As I said, it's not that the whole DNA opens up, only 10 to 20 bases open up. So nucleotides are added to the 3' end of the growing RNA polymerase. RNA polymerase works in 5' to 3' direction. So this is showing the elongation process. You can see this is the double-stranded DNA which has opened up, and the RNA polymerase is actually adding the ribonucleotides here. Okay, so they are being added at the 5' end. So only this much part of the DNA is opened. Okay, so this is your enzyme RNA polymerase. This is opened DNA. Okay, and this is the template strand of DNA. Okay, termination of transcription. So there is a difference in termination in the prokaryotes and the eukaryotes. In the bacteria, the prokaryotes, the polymerase stops transcription. In bacteria, that polymerase stops transcription at the end of the terminator. In bacteria, the polymerase stops transcription at the end of terminator, and the mRNA can be translated without further modification. But in eukaryotes, RNA polymerase 2 transcribes the polyadenylation signal sequence. The RNA transcript is released 10 to 35 nucleotides past this polyadenylation sequence. So in the eukaryotes, there is a signal called polyadenylation signal, and the RNA polymerase transcribes the polyadenylation signal sequence, and it basically goes beyond that, and the RNA transcript is released 10 to 35 nucleotides past this polyadenylation sequence. Okay, as we said, the processing also takes place in the eukaryotic transcription. There are three steps in the processing which takes place. One is the addition of the poly A tail, which takes place at the 3' end, and there is also addition of 5' cap on the 5' end. So mRNA has two ends, 5' end and the 3' end. So on the 3' end is addition of a poly A tail. So what's a poly A tail? It's basically the it's a stretch of A's end. What is A? It's adenine. Okay, so there is a on the 3' end there is addition of adenines, and it forms a kind of a tail. So it's called a poly A tail. And on the 5' end there is addition of a nucleotide which is a modified form of guanosine, which is added on the 5' end of the mRNA. So why are the two ends of mRNA modified? The two ends of mRNA are modified to make mRNA more stable. Why? Because mRNA is very sensitive molecule, and mRNA has to move from nucleus to the cytoplasm, and on the way there are many enzymes, and those enzymes can degrade mRNA. If mRNA is degraded, then no protein will be formed. So that is why, to make mRNA more stable, the two ends of the mRNA, they are modified. So this is basically the poly A tail. Okay, 50 to 20 adenine nucleotides added to the 3' end. So this is the 3' end. Here 50 to 250. So this is the 3' end. On this side, 50 to 250 adenine nucleotides are added here. And this is the 5 prime side. A modified guanine nucleotide is added to the 5 prime end. And basically, in the mRNA which is made, there are some regions called introns and there are some regions called exons. Introns are basically a kind of a junk DNA. Okay, so exons are the ones which codes for proteins. So introns, or you can remember introns from something which is interfering. It's also called junk DNA. So remember that we're talking about mRNA, but that information in mRNA is coming from DNA. So the part of the mRNA which is coding for an intron is coming, that information is basically coming from DNA. That information is coming from DNA. So there are introns and exons in DNA, and when they're coded in mRNA, those exons, they are removed by a process of RNA splicing. We are going to talk about that, and that takes place with the help of a complex of RNA and proteins that's called spliceosome. Introns are removed and all the exons, they are joined together. Okay, so I think it's shown here. It's shown very nicely in this picture. So this is the your mRNA. Here 5 prime cap is attached. Here poly A tail is attached. You can see that this is light part is, this is intron, this is intron, this is axon, this is axon, this is axon. Okay, all the dark parts you see, these are axons, and all these light parts you see, these are introns. So during splicing these introns, they are removed and all these axons, they're joined. So you can see this is all coding segment. Okay, so so this is the mature form of mRNA. Okay, this is not a mature form of mRNA. This is a mature form of mRNA, because in this there's a 5 prime cap, there's a poly A tail, and all introns they have been removed, and the axons, they have been joined. And you can see when all the axons they have been joined, they form the coding segment. And this is the mature form of mRNA. And also there's important thing to know in the mature form of mRNA is that on the 5 prime side there's a 5 prime UTR, and on the 3 prime side there is a 3 prime UTR. Now what are UTRs? UTRs are untranslated regions. Okay, so we don't know why mature form of mRNA they have. We don't know their function yet, but they may have some role in some kind of regulation, but they are there in the mature form of mRNA. So this is the structure of mature form of mRNA. As I said, as I talked about RNA splicing, which takes place with the help of spliceosome. What's a spliceosome? It's a complex which is made of RNA and proteins. So you can see this is a spliceosome. This is a spliceosome, which is a complex of it's a complex of proteins. So this is a protein, and this is a complex of RNA and proteins. Okay, so these are actually called sRNPs. These are small ribonucleoproteins. So this is a complex of RNA and protein. So these are all SNRNPs, SNPs, sRNPs, small ribonucleoproteins. So they are complex of RNA and proteins, and they are further complex with other proteins. So that forms a spliceosome. And the function of this spliceosome is to remove the introns, and it helps in the joining of the axons. So this is, you can say, intron is removed, and the axon is joined together. Okay, so basically, how does splicing takes place? It takes place with the help of these spliceosomes, and in these spliceosomes, there is a RNA, and this RNA acts as a catalyst. This RNA acts as an enzyme, and when RNA acts as an enzyme, They are called ribozymes. So ribosomes are catalytic RNA molecules that function as enzymes, and they can splice RNA. Now these are some of the properties of RNA which enable it to function as enzyme. RNA can form a three-dimensional structure because of its ability to base pair with itself. Some bases in RNA contains functional groups that may participate in catalysis. RNA may hydrogen bond with other nucleic acids. So these are some of the properties of RNA because of which RNA can act as a catalyst. Okay, there's another process which is called alternative splicing. Alternative splicing is, let me explain you from this picture. So basically, if this is a protein, if this is your mRNA, and it's shown here as a DNA or a gene because the information or whatever is there in the mRNA is actually coming back from the DNA. So if this is one exon, this is intron, exon 2, exon 3. So during the transcription, these introns will be removed and these exons will be joined. Now it means that in the mature form of mRNA, there will be exon 1, exon 2 and exon 3. Now exon 1, exon 2, exon 3, how they are arranged? It can be arranged exon 1, 2 and 3 together. That forms one kind of a protein. There's a possibility that after exon 1 comes the exon 3, then comes the exon 2. Then it will be like exon 1, exon 3 and exon 2. Okay. Then it will be like exon 1, exon 3, exon 2. It is going to be a different protein. Or maybe it can be exon 2, exon 3, exon 1. It's going to be different protein. So how these exons they are arranged, they form, on the basis of the arrangement, they form different proteins. So this process is called alternative splicing. Okay, this process is called alternative splicing, which means how these exons they joined, it can be in a very alternative pattern, and because of that different kinds of proteins can be made. And basically every exon codes for a domain. Now what is a domain? Domain is that part of the protein which can domain is that part of the protein, of a fully folded protein, which can perform an independent function. That is what a domain is. So domain is that part of a fully folded protein which has attained a three-dimensional structure which can perform a independent function. Okay, that is what a domain is. And every domain is coded by a axon. So this is what is being shown here. Okay, the next part is translation, and I'm going to make a separate video for that.